Ms. Sara, and my topic of this research paper is of the ethics and technology behind animal cloning. The purpose of this paper is to discuss these issues of cloning and technology. Animal cloning is made up of many processes to produce an organism that is genetically identical to the original. Many Americans claim to be morally against animal cloning, yet there is a lack of public discussion. Similarly, animals have been used as test subjects for many kinds of lab experiments. Their health and well-being are often disregarded in these aspects. Cloning has had an impact on the agriculture and pharmaceutical industries, such that sheep have been genetically engineered to produce a human protein for the sole purpose of blood clotting as a medical treatment. Cloning animals enable scientists to genetically modify cow meat to be more nutritious, but it could also possibly aid in reviving endangered species. The three types of artificial cloning are therapeutic cloning, reproductive cloning, and gene cloning. In therapeutic cloning, embryonic stem cells are produced to fix damaged tissues. Gene cloning produces replicas of segments of DNA, and whole copies of organisms are created through reproductive cloning. Pet cloning is a debate on its own, considering the expense and unnecessary pain it causes to many animals. Exact replicas are also not promised. Cloned animals live a short life and possess many health problems. The success rate of cloned animals is very low, so many surrogate animals as well as deformed clones suffer. If the success rate of cloned animals is high in the future, it is possible that humans will be the next test subjects to be cloned, and this would cause an outrageous ethical dilemma. Public discussions on cloning need to be discussed in terms of health, morals, and religious beliefs. In 1996, scientists achieved the first successful animal clone by utilizing an adult sheep's somatic cell, which is a non-reproductive cell, from her udder. The researchers went through numerous trials until they created the cloned lamb named Dolly. Cloning consists of a multitude of various processes that are utilized to produce a genetically identical replica of a living animal. Scientists have used cloning techniques with cells, tissues, and genes along with whole organisms like Dolly. This was considered groundbreaking research that continues to be improved upon today. Scientists have successfully created clones of other species, which include dogs, rats, cats, rabbits, deer, and chicken. Regardless of this research, the ethics behind animal cloning has not been fully addressed by the public. Animals have been used as test subjects for many types of scientific research. The interesting matter of debate is that 64% of Americans hold negative views against animal cloning, yet there is a lack of discussion and protest for change and regulation among the public. Without discussion, it's difficult to produce a resolution for this type of animal biotechnology. A reason for the inadequate discussion on the ethics of this issue may be due to the general view of animal rights and the treatment they experience. They experience traumatic circumstances as test subjects in evaluations of eye irritancy, dose toxicity, skin irritation, and numerous other experimental health effects. These tests contribute to the efficacy regarding certain chemicals that are used in products such as cosmetics and household items. Cloning has been used in various areas such as the agriculture and pharmaceutical industries. One kind of genetic alteration consists of the creation of transgenic animals. These animals possess genetic material from a distinct species that has been artificially inserted into its original genome. Sheep have been created to secrete factor IX, which is a human protein that helps promote blood clotting. Retrieving this protein from sheep milk can help people with hemophilia. This information was referenced from the article Ethical Issues in Animal Cloning, written by Feister. Animal cloning is mainly used to benefit the human species, whether it be modifying genes for more nutritious meat, cloning disease-resistant animals, or reducing environmental burden. For instance, researchers are in the midst of cloning chickens that are featherless to diminish poultry farming's environmental expense. Animals have been cloned for entertainment purposes as well, like genetically modifying a buck to have bigger antlers that appeal to hunters for personal decoration. Animal cloning has also been used to try to benefit endangered or extinct species. Therapeutic, gene, and reproductive cloning are the three types of artificial cloning that exist. Embryonic stem cells are produced through therapeutic cloning, and this process remedies unhealthy or damaged tissues. Replicas of segments of DNA or genes are formed through gene cloning. Copies of organisms are created through reproductive cloning. The method of gene cloning consists of injecting a certain gene from one organism into genetic material of another organism's carrier. The carrier is known as a vector, and common vectors are yeast cells, viruses, and bacteria. Following the insertion, the vector is situated in conditions that encourage it to multiply. This produces multiple gene copies. Mature somatic cells, such as skin cells, are removed during reproductive cloning, and the DNA of the animal is relocated into an oocyte, or egg cell, that previously had its original nucleus removed. DNA from the mature somatic cell is added to the egg by removing the DNA-containing nucleus and inserting it into an empty egg, or by fusing the egg and the cell by means of electrical currents. When this process is complete, the female animal births offspring that possesses identical genetic material as the somatic cell that was implanted inside. This is known as a clone. This information was referenced from the website genome.gov. Pet cloning is another type of cloning that has its own ethical issues. It seems to be targeted towards wealthy people since a single person would have to pay $25,000 to $50,000 for a clone. And this was referenced from the Forbes article named "If You Love Animals, Don't Clone Your Pet," written by Barron. Some people cannot cope with the loss of losing their pet, so that they use this as a reason to purchase a clone. Unfortunately, it is impossible to exactly clone an individual cat or dog. The cloned animal may look similar to the original, but it will never look or act exactly the same. Fur coloring as well as personality would be different, since it is impossible to bring back the soul and transfer it to a clone. There is also the issue of the number of surrogates that is required to create a clone. Considering that producing clones takes many attempts and many surrogates, details pertaining to the treatment of these surrogates and failed clones that have deformities remain in the dark. Cloned animals are often sterile and possess many unexpected health issues, so they are unable to live average lifespans of regular animals. Many cats and dogs awaiting a loving home in shelters is used as an argument against pet cloning. Due to overpopulation, many healthy, adoptable animals are euthanized, so the cloning process brings more animals into the world to suffer, which raises an ethical issue. Animal cloning suggests the potential for cloning humans. Human cloning uses therapeutic cloning as a reference to cloning embryonic cells for healing damaged nerve and tissue cells. Reproductive cloning is similar with the utilization of the transfer of somatic cells. Cloning exact individuals remains impossible, but it is possible to clone genes. The only natural cloning that takes place in humans is a mother giving birth to twins. And this was referenced from the article "Cloning Humans: Biological, Ethical, and Social Considerations," written by Ayala. Animal cloning has proved that the clones do not live a very sustainable life, so it would be unethical to try these procedures on human beings. Cloned animals suffer through organ failures, distorted limbs, shortened lifespans, and many other traumatic circumstances. It is possible that cloning could advance in the near future, and it is possible that cloned organs may turn out to be successful. However, the limitations are only to cloned genes. Personality is not assessed by the person's genotype. Regardless of whether cloned human beings would be born at different times or the same time, the clones will not be behaviorally or physically alike with one another at the same age. This information was referenced from Scientific and Medical Aspects of Human Reproductive Cloning book's website. Animal cloning consists of many ethical issues. Currently, the success rate of perfectly cloned animals is extremely low. While clones of animals may have the potential to benefit humans in the future, such as creating nutritious meat or creating disease-resistant animals, it is imperative to do more research and justification of experiments and weigh out the advantages and disadvantages of the case. Is it moral to use technology to breed animals into existence when their lives will be cut short due to genetic defects or sudden health implications? That is a debate that needs to be further discussed in order to continue to use technology for this purpose. If the goal for cloning is to make humans more disease-resistant or live a better life, then cloning could help in terms of the quality of life. The cloning of humans in itself presents significant moral, religious, and medical consequences. that must be addressed by the public and not just by scientists, politicians, and others that may not be motivated by factors that do not address this ethical dilemma.